In a previous video, we discussed the digestion and absorption process of protein molecules. In this video, we will be discussing the same digestion and absorption process. However, this video will entail the discussion of carbohydrates. Okay? Now, the two main processes that occur within the entire digestion process include mechanical digestion as well as chemical digestion. Now, in the case of proteins, only mechanical digestion occurs. And we know mechanical digestion, its purpose is to increase the surface area of food molecules for enzymes to better work at them to break them down into smaller units, okay? So chemical digestion is the utilization of these proteolytic enzymes that break down food molecules into smaller units. Now, in the case of carbohydrates, both mechanical digestion, right, as well as chemical digestion occurs in the mouth, okay? Now, the enzyme that starts the breakdown of these carbohydrate molecules is something called salivary amylase. Salivary, obviously referring to saliva. Amylase, right, is an enzyme that breaks down carbohydrates. Okay, so whenever you see the suffix ase, okay, that will most likely refer to an enzyme, and amylase, okay, refers to the breakdown of a carbohydrate. Okay, now, what is a carbohydrate? Put very basically, a carbohydrate or a polysaccharide, right, we'll use that example, is essentially a chain of smaller sugar molecules. For example, glucose. Okay, now the links, okay, between these glucose molecules are called alpha glycosidic bonds. The enzymes within the human body can break these alpha glycosidic bonds. However, there are some carbohydrates that are linked, the sugar molecules are linked via beta glycosidic bonds. Our body's enzymes cannot break those down. Okay, now what carbohydrates have these beta glycosidic bonds? Cellulose, fiber, for example, right? Our body can't break down fiber, but it can break or cleave these alpha glycosidic bonds via a process called hydrolysis, okay, with the help of these enzymes. Now, so we ingest carbohydrates, mechanical digestion and chemical digestion occur in the mouth, breaking them into smaller based polysaccharides. Saccharides refers to sugars, poly refers to many, polysaccharide, okay? Now, the next stop in the digestion process is obviously the stomach via the esophagus, okay? Now, chemical digestion of carbohydrates doesn't occur in the stomach, okay? The molecule that does break down into smaller units within the stomach, as discussed last video, is protein molecules. So the next step in the digestive canal, okay, is the lumen of the small intestine. Specifically, the first segment in the small intestine, which we know is the duodenum. Okay, there's three segments within the small intestine: the duodenum, the jejunum, and the ileum. Okay, absorption primarily occurs in the jejunum and the ileum. Digestion primarily occurs in the duodenum. Okay, so once polysaccharides, once they enter the duodenum, we have hormones that are released from the small intestine, namely cholecystokinin or CCK, as well as secretin. These two hormones stimulate the pancreas to release enzymes called pancreatic amylase. There's that word, amylase again. So it breaks down these polysaccharides into disaccharides. Again, saccharides referring to sugars, di referring to two. So it's breaking it down into smaller units. Now, these carbohydrates, they then come into contact with enzymes at the brush border. Now, the brush border is essentially finger-like projections, right, called villi and microvilli that break down food molecules, and the topic of this conversation, obviously carbohydrates. It breaks them down into smaller units where they are then absorbed. Now, the enzymes that are present at the brush border are called disaccharidases. Okay, again, saccharide referring to sugar, di referring to two, ace refers to an enzyme, okay? So specifically, these disaccharidases that are present at the brush border are called maltase, sucrase, and lactase. Maltase breaks down maltose, which is two glucose molecules connected together. Sucrase breaks down sucrose, which is a glucose and a fructose attached together. Lactase breaks down lactose, which is a glucose and a galactose attached together. So it breaks them down into these monomeric units, these simple sugars, galactose, glucose, fructose, okay? Now, here is the absorption process, okay? So again, digestion primarily occurs in the duodenum of the small intestine, whereas absorption occurs in the jejunum and the ileum, okay? So that's mostly where this process takes place. Now, fructose, it passes down into the enterocyte passively, okay? And as a reminder, an enterocyte, entero refers to intestine, right? Or digestion. And cyte refers to cell. So the enterocyte literally means, right, intestinal cell, okay? So fructose, it passes down into the enterocyte passively via this transmembrane protein, right, to help it get through. Now, before we discuss this, we have to talk about the sodium-potassium pump, okay? Again, if you want a more detailed explanation of the sodium-potassium pump, you can look it up on YouTube. But put simply, it pumps 3 sodiums out of the cell and 2 potassiums in. So it creates a higher concentration of sodium on the outside of the cell compared to inside. And we know that molecules in general in the body, it's their tendency is to go from a high concentration to a low concentration, right? And that's not just specific food molecules. That relates to oxygen, right? Carbon dioxide, water, right? Which is the process of osmosis, goes from a higher concentration to a lower concentration. So if we have a higher concentration on the outside of the cell, this is the basolateral side, then sodium will want to come from a higher concentration on the outside, on the apical side, that's this side, the side towards the lumen, to the inside of the cell, right? So this membrane, this protein right here, it's called a symporter, meaning that more than one molecule is coming with it, right? So sodium is coming down its concentration gradient, and it brings glucose and galactose molecules with it, right? So these glucose and galactose, those are called 6-membered sugars. Fructose is a 5-membered sugar. It goes down its gradient passively, whereas glucose and galactose come with sodium, right? Sodium-linked secondary active transport, okay? Or just simply sodium-linked dependent transport, okay? Now, fructose is converted into galactose within the cell, and these simple sugars go down their concentration gradient into the bloodstream where they're absorbed by the body. They can be taken to the liver to be converted to glycogen, right, into these complex carbohydrate units, or to the cell, right, where they're converted to glycogen, or these sugars can be converted to fat and put into adipose tissue, okay? So this is the digestion and absorption process of carbohydrates.